Our professor who's here today is named Aaron Wright, and Aaron came to Baylor a couple years ago from a national lab in Washington State and Washington State University, and he does microbiome research. Do any of y'all know what microbiome is? I'll be honest, that was something I heard a little bit later in life, but when I heard it was also part of the gut. It dealt with the gut, what's going on in your stomach. Well, then it made a little more sense to me. And that's where Aaron Wright, where his research lives in a lot of ways. The processes that are going on in your stomach that impact our health and how we feel and how what we eat and the environment around us impacts our overall health. And I'm going to let him come on up here because he can explain it a whole lot better than I can. But could you give a little round of applause for Dr. Wright?
I find the right place to stand so you don't have to see my bald head in the light. All right, so we don't make people fly. So we said that, but I don't think we're that far away. We do a lot of experiments though, so I'm going to talk about the kind of things we do and how it relates to you, and then I'll try to relate some of our science, the type of experiments we do, to what you're going to do with us today with the lab stuff. All right, so we're going to talk about gut microbes in a minute. I'll tell you where I came from, just to give you a little bit of sense. I grew up in the Seattle area of Washington State. Went to the school that used to be the only high school that had a kangaroo for a mascot, and then until Colleen decided they were going to also have kangaroos. Yeah. And then I got a degree in chemistry from a small little university in Oregon called George Fox. Then I moved to Texas the first time. I went to the University of Texas and stayed in chemistry and got a doctorate degree in chemistry. Then spent some time in San Diego at a place called the Scripps Research Institute doing research. We've done a lot of research. And then on to the national lab, and now at Baylor for the last couple of years as a professor. So it's been a long course to get here. The national lab was back in Washington State, so my wife and I, my wife's here, sitting down as a teacher. We've been back and forth to Texas and Washington a couple times. All right, so if you're curious, what does it mean to be a professor of biomedical sciences? I teach a little bit, right? Some professors teach a lot, I teach a little bit. I mentor a lot. There's 3 of my graduate students are over here. You'll hear more about them from them later. And then I do research, and I do a lot of research. This is kind of the main thing that my group does, is that we focus on a research project where we feel like we can help impact people, science, and health. So there's a lot of students, it's probably kind of hard for you to see this picture, there's a lot of students that work with me, and they're in our group, the Wright group. And these are the kind of things we do. And don't worry if these don't make any sense to you, because we're going to talk about it. First of all, we work on gut bacteria. And gut bacteria is inside of your intestines, lines your intestines, it's moving through your intestines, and when you poop, it's full of bacteria. And so myself and these 3 scientists over here, we are all officially poop scientists, because that's where most of our research gets done at, is actually working with poop samples, or mouse. A lot of mouse work and open up their intestines. And that bacteria does some really important things for us. The things that you don't even think about, of course, because you're probably not worried about the bacteria that's inside of you. There's a lot of bacteria inside of you. As much as 2 to 6 pounds of bacteria can be inside of an adult human, so that's high. Most of you probably are 1 to 2 pounds of bacteria living inside of your stomach. And so if you wonder, well, stomach and intestines, and if you wonder why, it's because they help do things. So we're interested in how do these bacteria inside of us that we call microbes, we call the whole environment the microbiome. We're interested in how do they help us process drugs, like medicines, therapeutic medicines. We're interested in what happens if pollutants, if we get exposed to those, how does that change? We're interested in like early life. So if a baby gets exposed to nasty environmental pollutants, how does that then affect their ability, their bacteria in their stomach later on? And then we're interested in diseases like irritable bowel diseases, like ulcerative colitis. Maybe some of you know people have Crohn's disease or ulcerative colitis, irritable bowel disease, these sort of things. They're dealing with problems in their intestines, and the bacteria that are in there are part of that. Now, for the people that like experiments, we do a lot of experiments. My group is both chemistry and biology. So we're making things using chemistry, we're analyzing things using chemistry, And then we're doing a lot of biology to try to measure the bacteria and what they're doing inside of us. Somebody in my lab made this. Maybe some of you are familiar with the Infinity Gauntlet. We renamed it the Chemomics Indy Gauntlet, which is sort of terrible, but it's fine. And this is just to show you that there's all sorts of different types of experiments we do. All this stuff is going on in our lab all the time. We're using different types of machines to make measurements of things you've heard of. DNA and RNA, proteins, all of these are things that we make measurements of because they help us understand what's happening in our gut. So what I want to do is spend a couple minutes just talking about this term. What is a microbiome? So it looked like a lot of you, this is your first time you've heard this word, or maybe it's the first time you're getting a chance to hear more about it. And the most simplest way to understand this is it's the entire collection of microbes, that can be bacteria, fungus, viruses, that live together in some environment. Right? So for our gut, we call it the gut microbiome. You can also have a lung microbiome. Right? We have a skin microbiome. There are bacteria in your skin right now that are helping protect you and doing other things that are valuable for you on your skin, just like that's happening in our gut. When we think about how much of this stuff is around, I'll try to explain this in a second. This is how many, somebody has estimated how many gigatons, so billions of tons, of carbon exist on Earth, and how much of that is humans. And if you see, down here in this little tiny corner is 2 gigatons of carbon. And then people are over here at 0.06 gigatons of carbon. It's not very much. It's a small amount. But bacteria are over here at 70 gigatons of carbon. So there's way more bacteria on Earth than there is animals and humans. Way more, right? And they're important. They're important for all sorts of things. So we see bacteria, for instance, in the ocean, right? And they have symbiotic relationships with things like coral and helping coral reefs, these sort of things. They're certainly an animal, like fish have a microbiome. Starfish have a microbiome. They all have bacteria associated with them that are doing things for them. If we think about plants, so if you go outside and you scoop up a big thing of dirt, inside of that is a lot of bacteria, and a lot of viruses and fungi, and those are helping for different reasons. They help fix nitrogen, all right, so they also help with things like plant growth. So, and the roots, if you ever pull up a plant and look at the roots, there's a lot of bacteria that are associated with the roots. Sometimes you'll even see like crystal and stuff, which is nitrogen, that is fixed by bacteria. That's why you see that there at those roots. And then there's this, which is what we call the built environment. It's the things around you. This came up a lot during COVID, right? Is touching a surface potentially a problem? In COVID, it was largely decided later on that no, that wasn't really a problem. You don't need viruses. But in this room, like we worry about things. I'm sure you've probably done the experiment maybe where you swab the door handle and then put it on a piece of bread to see how much stuff grows. Have you guys done this experiment? No? Oh, you should do that experiment. Yeah. Or you can swap like the drink cooler, like the water cooler, these kind of things, and it's amazing how much bacteria exists. Well, that's people trying to understand the built environment. How do bacteria live in these places around us? How do they live on your stainless steel door handle? Maybe more importantly is how they live inside of a hospital. How do nasty bacteria, pathogenic bacteria, how do they end up inside a hospital? How do they end up inside the ductwork? Are bacteria and viruses moving through the air system? People worry about these things. So this is called the built environment. So I'm going to spend just the last couple minutes, though, talking about what's happening inside of you right now. Yeah? Oh,
no, just waving down this. No.
All right. What's happening inside of your intestines? You are made up of about 30 trillion human cells, and inside of your gut is about 38 trillion bacterial cells. But it takes about 10,000 bacteria to equal 1 human cell. Bacteria are very tiny. And these are the kind of things that they do for you. These are the kind of things they do for you. Right now, there is bacteria in your nose, and they're helping these things. Mucus production is one of those things that they maybe are associated with. They're in your mouth, right? Cavities. How many of you have had a cavity? Yeah. Behind that are pathogenic bacteria that aren't helping in that process. You should brush your teeth. It seems grosser when you're like, oh, cavity is actually like bacteria rooting away at your teeth. Yeah. Lungs, they help in your lung health. The stomach, lots of stuff going on. The colon is down in our intestines. The main thing that we think about, like in my group, is we're worried about how they help us digest things. So the food that you take in, there's a lot of food that you can't actually digest or get the benefit from until bacteria help us with that. And so a lot of that is carbohydrates and fiber are being, you're not able to ingest all of that, so they're helping break it down so that our bodies can actually use it. Okay. I'm not going to talk about this stuff. We're interested in lots of things.
Y'all know what any of that is when you see those pictures?
No? All right. Let's go to the next one. I'll show you some
You
of
can
these.
tell them, I mean, yeah.
Oh, I'll show you
For
some of
real.
these ones though. So,
Hold on.
Yeah, the things that we made. So do you know what you're looking at? So, okay, so that's what he's gonna show us, right? So those are the compounds, right? When we studied the elements and we built the molecules, that's just a different way to represent it than our chemical formulas.
yeah, so you guys probably, the easiest thing that we think about chemicals is things that you might have taken for like a headache, right? How many of you take like ibuprofen this week or an Advil, right? Those are chemical molecules. They're drawn like this. This is how chemists, we draw them to understand their arrangement of atoms. So how does the oxygen link into carbon? When we get more advanced drawing like this, we don't draw the carbons anymore. The carbons just become little spots where there's a peak, right? Where you point at the intersection. So that's all we're looking at here. So some of these things are actually important, like dietary compounds, the things that you're taking in. These are what some of these pollutants look like. And these are like some of the, I don't remember actually which drugs these are, but these are pharmaceutical drugs that people may be on for things like cardiac health. I think this one is digoxin, which is for heart health. And to the bacteria inside of us, and the reason why I put this up here, is that they can be both good, beneficial, and they can also have negative consequences. So they can help us metabolize things, but they can sometimes also make things more toxic. So sometimes when we take in, maybe we somehow are exposed to like a bad chemical, they may actually make it worse by doing some chemistry on it, and that makes it worse for us. One of the projects that we worked on a while ago, and I mentioned to show this real simply, is can we help doctors make better decisions? So one of the problems is that bacteria inside of your intestines can actually lead to some drugs not working very well. And one of the main problems is colorectal cancer and liver cancer. And they know this, they know that the bacteria cause problems to these drugs. And so, but it's not in everybody, it's only in certain people. And so we wanted to be able to figure out, can we actually determine which people are maybe not going to respond very well to those chemotherapeutics? And if they don't respond well, it delays their treatment when they should be getting a therapeutic, a cancer therapeutic. And so we wanted to see, could we help them? So we wanted to work in poop samples from those patients, right? Can we then come up with a way to make a measurement that allows us to determine whether or not a person may not respond well? So here's a whole bunch of examples. We had a whole bunch of poop samples given to us. Only one is actually from a cancer patient, and it's from a cancer patient who didn't respond well. And we wanted to know was, if we look at that person that didn't respond very well, do they have the bacterial protein in them at a high level that's causing that drug to not work very well? And that's what we found. It's actually one of the highest levels is this patient who's 61 years old, and they have a high level of this. So that's one way that we're trying to help inform doctors to make better decisions.
Was that the cancer patient?
Yeah, the one with the green box. Yeah, who didn't respond well. They get, in this case, it's a drug called irinotecan. They get really horrible diarrhea, and then they can get like, it's so severe that they have to take them off the medicine. It's almost a month sometimes to get them on new medicine. All right, last two slides, making a huge change to be related to what we're doing today with you. In our science, in our experiments, we do a lot of chromatography, right? So I've given you this big high-level idea of what we work on. We work on gut bacteria. But in day-to-day, the students are working on making things, on analyzing things, working with bacteria so they can actually make measurements. One of the things we do is called thin layer chromatography. And we do a lot of chemistry. So we make, like some of those molecules I showed you a couple slides ago, we make things like those. And so when we run a reaction, so Sam, wave your hand, and Lorraine have both done chemical reactions in the lab. When they mix two things. They might mix A and B, and they're hoping to make C. So this is kind of like putting together like baking cookies, right? But you don't always get perfect cookies, right, at home. Well, we don't always get perfect reactions that only make C. What usually happens is we mix A and B, and we get some of what we want, and then we get a whole bunch of other things that we don't really want, D, E, F, G. And you have to get those out of there. You have to find a way to remove them. And so we can analyze this happening during a reaction. This is thin layer chromatography. Every spot is a different chemical compound. If we were doing a reaction, we would hope to see 1, not 3. So now we know we have to separate things out. And so then we use more advanced techniques. This is a chromatography system where you have a big column over here. This column is packed with something called silica. And different chemical compounds move at different speeds through that silica. So if we take our C, D, and E and we load them on here, this thing separates them out, and now we get only C. And so we're doing some of that today with you all. All right, last slide. Again, back to the group. This is all the members of the group, or most of the members of the group. I'm going to let the 3 students here introduce themselves to you, and then because they'll end up at tables with you all, I will too, and some of the others here. So, Amelia, if you can start.
Hi everyone, my name is Amelia. Like Dr. Wright said, I'm a graduate student in the Wright lab. My work specifically is focusing on how psychological stress and also vitamin B12 are interacting with your gut. An example of how I use chromatography in my research. So to analyze how stress and vitamin B12 are interact with the gut, I went to measure bacterial products. So Dr. Wright mentioned that they do these chemical reactions, and so some of these products from these reactions I'm interested in. So you are going to use water for your chromatography reaction or experiment today. There's also something called gas chromatography. So instead of having the water be what pulls the compounds through, you have gas instead. So I use gas chromatography to separate some metabolites out and to study those and what I did.
How many of you before an exam or a quiz or something have ever had like you're stressed out or anything, maybe it's like a sports thing, and you feel really stressed and your stomach starts to hurt? Yeah. You ever thought about why is that? Why is my brain connected to my stomach? That's what Amelia's working on. Yeah. Why does your stomach hurt when you get nervous? Yeah. Go ahead. This is Ray.
Yes, my name is Ray. I'm a third-year grad student in Dr. Wright's lab. I'm also from Washington State, like Dr. Wright. And I'm here to study dietary things, and specifically like artificial sweeteners. So if any of you guys have diets, Coke or gum, there's something in there that's kind of like fake sugar. And so I look at how those fake sugars can impact the gut bacteria and change the way that they behave. And yeah. I also use gas chromatography, like Amelia stated, so we're really interested in what bacteria are capable of and how different factors impact them. Yeah.
Ray's from Washington, like you said. Amelia's from Maryland.
I did.
That's why she says water, Tony. That's Dan.
I talk really funny.
Yeah.
So, my name's Sam. I'm from England. I'm a second-year chemistry PhD student. And so, while Amelia and Ray are biologists, the work that I do is kind of focused more on actually making the molecules that we call probes that Dr. Wright talked about. So, when we're looking for specific enzymes, that might be doing specific things in the gut microbiome, the proteins in there, we have to make different molecules to go after different enzymes. And so really kind of what I do is like the chemical synthesis of sort of like different baits. If you think about going fishing, if you're fishing for different fish, you might use different baits. And so I try to synthesize different molecules that can go after different enzymes. So that's the work that I'm really focused on. Types of chromatography I use are basically what Dr. Wright presented, the thin layer chromatography, and what we call semi-preparative chromatography, which is where you can actually purify out the molecules that you've made. Running real TLCs and all that to like make sure we smart on the plate once you switch over. So you guys can do some other ones. The solvent front is cool when
Luckily it's just you.
The furthest that it got. 7. The furthest that it got. Those two ones. They're color spread on right now.